Welcome to Daily Series, COVID-19 Special. What do we need to know about the new vaccines? What is mRNA? And what we can expect from the first batch of COVID-19 vaccines. mRNA technology, leading the pack in the COVID-19 vaccine race. Pfizer BioNTech has concluded a Phase 3 study of its vaccine, BNT162b2. Primary analysis has shown it to be about 95% effective against COVID-19. On the basis of these positive results, the United Kingdom granted emergency approval for the vaccine and began inoculating its citizens on December 8. In the United States, the U.S. Food and Drug Administration (FDA) and most indicators suggest that the vaccine will be authorized for emergency use shortly after an FDA meeting on December 10. Moderna has presented preliminary data from its Phase 3 trial. The results so far have shown that its vaccine candidate, mRNA-1273, also has an efficacy rate of about 95%. Emergency Use Authorization (EUA) for the Moderna vaccine is expected to come hard on the heels of an approval for Pfizer's vaccine. How does mRNA technology work? Both Pfizer and Moderna's vaccines rely on mRNA technology, which stimulates the body's immune system by using a genetic molecule, RNA, ribonucleic acid. RNA is similar to DNA and plays various vital biological roles, including the regulation and expression of genes. The University of Cambridge explains that an RNA vaccine works by introducing an mRNA sequence, the molecule that tells cells what to build, into the body. The mRNA sequence is coded for specific molecular structures on the surface of a virus, called antigens. The genetic coding instructs human cells to produce components of the targeted virus. These parts can't cause infection like the actual virus, but their presence stimulates a person's immune response. As you have the genetic information of a virus or a pathogen, you can begin to develop a messenger RNA vaccine against it. There's no infectious material being injected into an individual. The technology is one in which you have the information that codes for the viral protein that you're interested in. This technology basically has the genetic information that will make this protein when you put it into a cell. Cells are going to recognize it as foreign, and they're going to mount a response against it. Addressing concerns. While these vaccines are a type of genetic medicine, assures the public that it poses no danger of harm or change to people's own DNA or genetic makeup. One of the concerns that scientists will be monitoring is how long-lasting the mRNA vaccines will be. Kuhn cautions that it's possible these vaccines may not be sufficient for long-term protection. We'll have to wait and see. Although studies have shown them to be safe so far, health authorities will also be watching for side effects. The companies have to be very clear about what the adverse reactions are going to be. The important thing is that they will be very transient adverse effects, and if they do persist for more than a day or two, then obviously you have to follow up. Potential future use. A great potential in mRNA vaccines, possibly in protecting against influenza. Although the majority of flu vaccines are currently derived from chicken cells, believes mRNA technology may one day be applied to flu vaccines. He notes that the flu virus poses challenges because there are so many different strains of influenza that are constantly changing. Ellipsis. You don't have as much variation in COVID-19 surface proteins as we do with influenza, and so the vaccine that's being produced right now is very responsive to the type of COVID-19 that is circulating in the human population. Traditionally, for viruses, we've either taken a virus and inactivated it and used that as a vaccine, or we've taken a virus and made it less infectious, had as it's attenuated, and made that a vaccine. Or we've expressed proteins that are on the surface of a virus and used those proteins as stimulants for your immune system. These would be purified proteins that would be injected. Ellipsis. The protein self-assembles into something that resembles the virus, but it doesn't have any of the components that allow the virus to replicate. So those are the standard, traditional vaccines. Ellipsis. The technology that Moderna and Pfizer are pushing right now is one in which you use the coding sequence, the information that codes for the viral protein that you're interested in. In the case of COVID-19, we're interested in a surface protein that we call the spike glycoprotein, spike for short. This technology basically uses the genetic information that will make this spike protein when you put it into a cell. And that information is encoded in what we call messenger RNA, mRNA. That's the vaccine, and it's packaged in a lipid nanoparticle for delivery purposes. That's absolutely correct. First of all, there's no infectious material being injected into an individual, you're only making a single protein. But it's the critical protein that your immune system will respond to. What will happen is, that lipid nanoparticle will be able to enter cells in your body after you've been vaccinated. And that RNA, the messenger RNA, will make a protein. Just like all the proteins your cells normally make. The only difference being that once it gets made, other cells are going to recognize it as foreign. And they're going to mount a response against it. This technology has been around for a few years. In fact, Moderna developed the technology initially against Zika virus. Ellipsis. In the case of Zika virus, there was this massive expansion and infection of people in South and Central America, and everybody was very concerned, and then the virus died off. Moderna had this technology but was never able to go to clinical trials because there was no Zika virus prevalent in the population. The COVID-19 pandemic is the perfect situation for producing a messenger RNA vaccine. Because it's very easy to produce in a large scale. Because it's synthetic. You don't have to grow anything in cells, which has been the traditional way that you produce vaccines. It's very easy, it's very rapid. As soon as you have the genetic information of a virus or a pathogen, you can begin to develop a messenger RNA vaccine against it, which trims off years of very difficult work that we previously had to do with the older vaccines. Don't forget to like and hit subscribe. Thanks for watching, see you on my next video.